The allele for red and white, as we just saw, being incompletely dominant. So you get, in other words, what they're saying here, they don't use the word incomplete dominant. They say that heterozygous individuals have pink flowers. But your understanding of incomplete dominance means that you realize, okay, so if you cross a red one with a white one, you get pink. That's incomplete dominance. It's classic incomplete dominance, right? So what you're also told is that the allele for tall is dominant to the allele for short. So that's the normal classical Mendelian dominant recessive situation, right? And then what the question says is, so what's the significance of the fact that the two allele pairs are located on different chromosome pairs? It tells you that they're located on different chromosome pairs in the question. Why is that important is what the question is asking. So what that means, of course, is that they're not linked. And what they're looking for there is for you to say that because they're not linked, the law of segregation applies in this situation. Mendel's second law, the law of segregation, applies. And I would go even one step further to say that during gamete, in other words, explain it, during gamete formation, that it is equally likely for either of one of the pair of alleles in this situation to combine with either one of the other pair of alleles in the situation. So it's equally likely for example, in this situation, for the red allele to combine with a tall allele, as it is for a red allele to combine with a short allele, okay, and so on. So knowing that, this is, it's not a linked condition, and so what we do is we end up just doing our cross as we normally would, and you get the full assortment of gametes. So I'm going to do the 8-step method with you for this because it's a really good one to work on. So the first thing is we're going to say that we're going to list the parent phenotypes. So parent, let me make this a little smaller. Control. There we go. It's perfect. So parent phenotype is the first thing we're going to look at. When you're doing this cross, and it says, the question says, there's a plant with pink flowers that was heterozygous in this, so it's got pink flowers. If it's got pink flowers, then that means it's going to be, well, we'll look at the genotype in a second. Let's just finish off reading the question, I guess. A plant with pink flowers that's heterozygous in respect of stem height. So in other words, it's tall pink because it's heterozygous in respect of stem height, but they've told us that stem is dominant, right? So this one is going to be tall pink, and it's going to be it's crossed with, it says, one that is white flowers and a short stem. So it's white, we'll call it short and white for short, okay? So what we've done is we've listed the parent phenotypes. It's important to write down those words, parent phenotype, and not just write down the two characteristics of tall pink and short white, because parent phenotype is it's like labeling in a diagram, and they treat this like a label, like I said, like a label diagram. So it says use suitable symbols to determine the genotypes of all the possible offspring of the cross. They're asking you to do the full cross here, and there's lots of marks going for this. So the second thing you're going to do is you're going to list the parent genotypes. Okay, so the parent genotype in this case, we've been told it's tall, but it's heterozygous. So that means that it's going to be big T, small t. And it's pink. And this was where I would say use, it's pink, so it's got a red one and it's got a white one. That's the way I would label it. And we're going to cross that with the other parent, which is short and white. So it's small t, and it's got, the only way it can be white is if both of the alleles are the white alleles. Okay, if it has a mixture, it's going to be pink. To be red, it's got to have two R alleles. The third thing we're going to put in then is our process of meiosis. And the only thing that you use for meiosis is you use arrows to represent what's going on. So 1, 2, 3, 4 different possible outcomes for this when we list what are called the gametes. Okay, so we're using the arrows to represent the fact that meiosis is occurring, and we're going to end up with 4 different gametes. The first gamete is going to be big T, C, big R. Yeah, you can, it doesn't matter which one comes first, but as long as you make sure that you represent all four of them and you put circles around them, they do like to see that. So big T is equally likely, because they're not linked, right? This is Mendel's second law kicking in. It's just as likely for big T to combine with, just give me 1 second. I just want to make sure that there isn't somebody knocking on the door trying to get in here. No, good. Just want to put up there we go. Good. Okay. So I think the sharing is back on. If not, I'll just make sure that it's back on. So share screen. Now, so we're in a situation where we've got to put in all these alleles then. And we have a situation with Mendel's second law, just as likely for big T to combine with C big R as it is big T to combine with C W, which is the white version, which is just as likely as small t combining with C big R as it is small t combining with CW. With this other parent over here, the only gamete that can be formed is there's only small t available. That's the only gamete available. There's only small w available. So this is the other gamete that's going to be formed. Okay. Next stage is fertilization. And what that means for us means we're going to do a Punnett square. So our Punnett square is going to be, we'll take the small t, so the one from the right-hand side, that's our gamete there, and we're going to cross that in a big four-section Punnett cross. So on the outside here, we're going to put up our four gametes. So it's big T, big R, big T, big W, small t with a big R, and small t with the W. And so then you just, do the fertilization version, and you would get big T, small t, C big R, CW. Big T, small t, CW, CW, or small t, CR, CW, small t, CW, CW. So you get four different equally likely parent or offspring genotypes, which is what we list next. And I know you've just listed them there, but my advice would be it's good practice to write down the list of the genotypes of the parents of the offspring as well. So genotype of the progeny of the offspring, and you can use either word, but just know that you do need to know what both of those words are. So we're just going to list them again. I'm not going to bother, but I would, if I was you, list them again here, and underneath that, you then put in the phenotype of the progeny. Okay, so phenotype of progeny. I'm sure your handwriting is going to be better than mine on this graphics tablet. So let's look at this. The phenotype, the physical appearance of this first possible outcome for the offspring is going to be tall because it's heterozygous and it's dominant recessive relationship. Whereas with the combination of R and W, you're going to get pink. But the second one, it's also going to be tall, but now it's got two white versions of the gene. So it's going to be white. The third one, again, it's going to be pink, but it's going to be short or dwarf. So short, pink. And then the last one, as we said, is going to be also short, but in this case, it's going to be white. So you get the last thing we need to put in there is then a ratio. So now you can see why it's the 8-step method. It's 1 is to 1 is to 1 is to 1. All of these are equally likely to be produced in the offspring.